ద్రాక్ష నా లిస్ట్ రాసుకోమని చెప్పు, ఒకసారి సైన్ చేసి. దీని మీద ఉపన్యాసం అడ్మిట్ అని కొట్టాలి, ఎవరన్నా జాయిన్ అయితే వస్తారు. అడ్మిట్ అని మీటింగ్ స్టార్ట్ అవ్వగానే మీటింగ్ అది కొట్టేస్తే మనకి అది కింద పడతాయి అన్నమాట. ఇక్కడ కూర్చొని చూస్తూ ఉండే కదా అవుతుంది, అది చూస్తుంటే అయిపోతుంది.
Hello.
A very good morning to respected Professor Vijayan sir. Our keynote, our special invitee, Srimonta Gayan sir, and faculty of our college and faculty from the neighboring colleges, dear student participants. We are now going to start with the technical session first, and we'll have the inauguration session later as our principal ma'am will join later. So now we will be heading directly into the technical session one, which will be delivered by Dr. Srimonta Gayan sir, Assistant Professor, EMBO Global Investigator, Infosys Young Investigator, Merck Young Scientist, Molecular Reproduction Development and Genetics, IISc, Indian Institute of Science, Bangalore, India. So now we have the privilege of listening to sir. Sir, I request you to please address our students. Thank you.
Yeah. So yeah, good morning to all of you. It's my um, yeah, yes. So I'm really excited to be here, and I think this is the first time I'm going to speak this kind of audience. So try to bear with me, and yeah, I'll keep it very simple. But if you have um, problem in getting into it, please stop me, raise your hand, and don't hesitate. So again, it's my pleasure to be here, and um, uh, yeah. So I'm thanks Pradeep and other members of the organization for arranging such kind of activities. It's very important for the students, and yeah, thank you all the organizing committees. So let's begin. So Pradeep, how much time? One hour. All right. So yeah. So today I'm going to talk about so basically my lab works on RNA immunogenesis and the stem cell differentiations, and major focus is the epigenetic regulation of gene expression. So I'll make it very simple. Yeah. So as so this is a kind of blastocyst. So these are these red cells are these are the inner cell mass, which are the pluripotent stem cells it carries. So these pluripotent stem cells actually at the later stage of development is going to differentiate to different germ layers and germ cells, and then followed by it's going to give rise to organogenesis and different lineage forms. Okay. And then these are the other cell lineage, like these are the founder, you can see these are the ectoderm cells, which are going to take the placenta. So this is a common blastocyst. So now, one is a very interesting thing, as I say, that from these pluripotent stem cells, so we have different nervous cells, circulatory system related immune cells, liver, kidney, and etc. etc. So now if you look, so all these cells have got the same DNA or same genome. But still, so they give rise to different cell types, and not only the different cell types. So when this early development, so these cells are, segregated from this state, specified. So they keep continuing that memory for the lifetime. Like say, I would say, which are the immune system cells. So they keep producing the immune cells. They do not produce to the other kind of cells. So they remember. So, but the very long-standing and interesting question is that they have all the cells almost have the same DNA or same genome. So how they maintain their cellular identity, right? So they are actually this thought that what I was going to talk about, that there are several epigenetic modifications. If you have heart, chromatin, nucleosome, histone, they are modifications, and also some modifications happen on the DNA, like such as DNA methylation, and also it happens on the RNA, such as RNA methylation, etc., etc. Right? So these are different covalent modifications have been found in the DNA of stones of RNA. So these actually thought acts like a stamp, or mark, which is propagated through the lifetime through the cell division. Like say, if particular cell time, you know, so these cells, so they have specific covalent this kind of epigenetic modifications, and those modifications actually through the cell division, so it is propagated. So it is thought that these act as the stand for the memory, and these modifications actually also regulate the gene expression. Basically, all these different cell types, they are expressing certain kind of transcription factor or certain kind of genes. So some genes are switched on in a particular cell types, and same genes might be switched off in another. But the question, as I said, that how they keep going, maintain that. So they are these different epigenetic modifications or regulations comes into the play to orchestrate or facilitate that throughout the lifetime of the. And then often, so as you say, the cancer is such a scenario, people have read of the cancer stem cells, so they are also, people have found there are these kind of epigenetic modifications, it's become aberrant. So there's people found their dictates. So that's also linked to the aberrant gene expression, and which might be one of the major driver or regulators of the cancer pathogenesis. So basically, so we have one genome, many cell types, and then the epigenetics from the stem cell differentiation stage, that the epigenetics programming begin. So that's the propagate for the lifetime. That how this belief that the cell identity is maintained for the lifetime, and especially the stem cells reproducing and proliferating. So that there also that it's very important. So with that, I'll move a little bit, uh, early stage, like say how the germ cells, differentiations happens, So these are the primordial germ cells. From there, gradually there will be spermatogenesis and oogenesis, and then there will be fertilization, and then this is a zygote, and then gradually, as I explained, the blastocyst and the further development will happen. So now here also, if you look, you will have seen, so there are huge epigenetic reprogramming happens in this process of oogenesis as well as spermatogenesis and in the early stage of you. So there are like say DNA demethylation and histone programming exchange or different layers of different histone modification. I'm not going to detail. So at this stage, even before starting the early development of stem cells differences, so from the gametogenesis starting beginning genesis or spermatogenesis, so they are also, so it is there. So basically, I'll come back when, like say, the germ cell is going to form this PGC. So they are going to complete removal of this epigenetic modification, and then during the oogenesis and gametogenesis and the early development, so it will be re-established, and finally it will be established in the stem cell differences. So this is during from the beginning of your development, from the gametogenesis itself. So these epigenetic modification or regulations, including the stem cell differentiation, has huge implications. So not only that, if you look at at this early development, so zygote to these are the different stages, 2 cell, 4 cell, 8 cell, and then this is the till the last process what I explained at the beginning of this. So there are several different cellular and molecular changes is happening here, and along with that you can see. So there are different. I'm not going to detail. So when this different lineage is segregating different stem cell is establishing, you can say man stem cell, plus stem cell, plus stem cells. So there are huge implications of these epigenetic drivers or the epigenetic modifications. Okay? So, and also what happens, so this is just very quick, two histone modifications called H3 lysine 4 methylation or H3 K27. So if you have this mark H3 K4 methylation, it's generally what it does in the promoter of the gene, it's active in the gene expression. And this mark, when we have the histone residue lysine 27, we have the methylation. So it's generally turn off the genes. So how you can instrument this chromatic mark. So if you have this again STK483, the gene remains active, keep transcribing, and if you have the STK2783, so a gene will turn off. There are exceptions. So now, so what happens? You can see from this different stage of development from the oocyte and the fertilization, zygote, two cell stage, eight cell stage, and then this is the blastocyst. So throughout these, so actually at the beginning you didn't have this mark very much and the gene was. So during this development, what happens gradually, as you can see from this late 2 cell to 8 cell, these are the pigments, so these are the promoter of the gene, and these are the blue peaks with the HTK483 mark is getting established. So it is also starting, so it was not there, neither you have it sign, neither the sperm, neither you at the beginning when you have the fertilization of zygote form. So it's just starting when the linear segregation is going to happen because from here you have different lineages and stem cell populations. So as you can see, as soon as the stem cell is going to establish, so it is just also starting to get established there. So as you can see, and then also when you have at this stage some of the developmental genes, basically which are very much important towards the differentiation of the stem cell at the early stage. So they then become marked by both of these mark. So basically they are prepared, like as I said, one mark will turn on the gene, another mark will turn off the gene. And then what they are basically doing, that's onset of stem cell differentiation. So they are preparing both of the marks. So now you might be confused. So if you have both of the marks, whether that genes will express or turn on, right? So yeah, they just express very minimally. So this we call that this voice state. So basically they are preparing during the stem cell differentiation because as I said at the beginning that after stem cell differentiation the cell identity how it's maintained that some genes get turned on in that particular cell, some genes is turned off and that has to be maintained. So now, at the beginning, so from these pluripotent stem cells, so they have capability to give rise to all kind of images, correct? So now, so they have to travel different diverse paths. So that's why they are getting prepared, as you can see, the basically the developmental gene. So they have kept both of them up because some cells that same gene has to be turned on and the another cell has to turn on. So now based on their requirement, like say a particular cell type has to turn on, what they is going to do, so they treat that this active part and then they get it on the inactive part. And similarly, in which cells they are going to differentiate, they going to keep inactivity, so they keep the inactive one and get it active. But as they have to do this very very quickly and carefully, very quick time window because you don't have much time, and immediately you have to specify two different cell types. So that's why they have choose this kind of phenomena that to keep both the market based on the requirement to get rid of one of them. And then the most interesting thing, as I started at the beginning, now when after this differentiation, the specific cell type is specified, so now they want to keep that gene on, so they are keeping the STK for any gene. So now throughout the lifetime, that looks like that one. So that's the most fascinating thing. So that's how these epigenetic modifications or regulations is instrumental from the onset of the very early development, and then it's specifying the cell type identity, and then it's maintaining in the lifetime. And when it goes wrong, you have different kind of diseases or disorder. And then as I said at the last slide, if you remember, that now when you are going to upward again for the next reverse. So for the jump sets, you are homogeneous. So in the end, you have the genome. This going to remove all this part and then looking at this stage, very early development steps and frequencies. Serious. So moving with that, so now I'll just touch upon it's a very, very interesting thing here, So far we have discussed, so this is the very this is two cell stage blastocyst, so this is continuous. So after that, what happens? These blastocysts will be implanted into the uterine wall. So before this implantation in the uterine tube, so this is like a protein wall, and then it gets during the implantation it gets attached to the uterine wall, and then this process called as implantation, and then a very interesting stage of development comes that is we call the pre-gastrulation and gastrulation. So pre-gastrulation, so I'll just very simply I'll explain. So this is a representative cartoon of a mouse or human embryo in general. So how the pre-gastrulation embryo looks. So as you can see, these green cells are actually the epiblast. So these are first the pluripotent epiblast stem cells, which actually comes from these blastocysts pluripotent stem cells. So upon implantation, these green cells become the pluripotent epiblast stem cells. And these epiblast actually down the line going to form all the different germ layers. If you have the mesoderm, ectoderm, endoderm, okay, and followed by the different lineage, different cell type will be formed, and then the organisms will happen. Okay, so now so here in this gastrulating embryo, as you can see, these are the cells, the external cells which basically form the placenta, yolk sac, which is very important for the development also. So, but I focus on these epiblast stem cells, and then what happens during this gastrulation? So, as I said, these epiblast stem cells are the pluripotent stem cells. Okay, so they basically have the capability to give the all kind of lineages from your body. Okay? So these pluripotent stem cells during gastrulation, what happens? So they start to very quickly. This is a mouse development, like 6 days of development. So they start to come out from the pluripotency and start to form these different germ layers. As you can see, I have colored different cell types. So these are the blue cells, which are the forming of the ectodermal cells, and this is the red cells, which are the endodermal stem cells, and then these are the yellow cells, mesoderm. So basically, the gastrulation is defined by these. So these pluripotent epiblast cells exiting from the pluripotency and quickly forming these three germ layers, ectoderm, endoderm, and mesoderm. And they followed by what will happen, ectoderm will give different lineages, endoderm will give, so that the entire embryo will form and organogenesis for the things will happen. So I'm not going further. And then at this time also what happens, some of these epiblast cells beyond these germ layers, they will form some germ cells, as I discussed in the previous slide. This is called the primordial germ cells. So those germ cells will be preserved for the next generation. So they will oogenesis to spermatogenesis. So they will undergo the gametogenesis. So now let's come back here. So now this is very fascinating, because what happens both in human and mouse very quickly. So you have one particular cell type, and then you are going to go different cell types very immediately. And not only that, now they are also have to segregate different number of different cell types. So that's why it is called, if you take a developmental biology textbook, you might find this interesting code. So basically this stage is highly dynamic. So these cells are undergoing a tremendous different transition, cellular state transition actually of the embryo. And another beauty is that it has to be very, very precise. Why I say these cells, three germ layers has to be like If you just give the ectoderm and ectoderm, don't give mesoderm, so actually embryogenesis will not be happening. So you have to give. Or not only that, you have to give the all. So you have to give very symmetrically all three terms, so that you have enough seed to form different organ from the stem cell. Correct? So that's why this is a very important step. This is the one of the renowned developmental biologists Lee so far. So he quoted that's very famous quote that generally what we consider in our lifetime, that some of the important time is the taking birth and or marriage and of course death. So what he quoted that I think much more interesting things is happening when you are masturbating at the stage of your life. So that's very dynamic, and so I think therefore he quoted the gastulation is the another like very exciting and important stage of our life which we not consider. So now come back to these. So now recently, so we will have, I'm not going to detail. So try to understand the advent of technology that these apical stem cells or these gastrulating embryo, these green cells. So these are actually one cell type, pluripotent cells. So down the line, they are going from the germ layers. And as I said, so they are going through the dynamic state transition. So recently people have identified, so these cells actually, so as you can see, these are the different from these cells. So different color patches. So actually they identified that it consists actually 30 to 40 different transient cellular state. So what is interesting here, in a cell type, so you can find in this stage of development, you have 30 to 40 different kind of cell types. Not exact different cell types, but they are very different. That's why you can see these patches. So this is one, these cells are very similar, and these cells are very similar, but they are very different from these cells. But they are all coming from the same cells. So that's so that there are within a cell type, you have different 30 to 40 states. So that is said you do not get if you take any other tissue or any part of the things. So many cells. You can get some difference of heterogeneity that all cells are not exactly if you could go in very depth with the technology. So you see that there are differences. But you never find such difference in any stage or in cancer cells or any other tissue or any other cell types population. So this happens only so far to the knowledge that these cellular cells, at least, so they are so many different cellular state, and that give the luxury, and plasticity or elasticity, so that quickly they can take different lineages, and so that the organogenesis of the things happens rapidly. Okay. So yeah, so now I just so this is what I have discussed basically that we say that from beginning of the gametes and then the fertilization, RDM, embryogenesis, that's basically giving the gastrulation from the stem cell to form the different lineages. So they are very much instrumental role in the epigenetic regulation that different histone molecules replace very important role. And this is the one of the biggest questions, very dynamic. Of course, so there are touching, there are different cellular signaling pathways, you have the and other factors. Of course, it's very important role. But what I'm going to very quickly introduce to you, you may not have been, so that the one thing how the cell is maintaining the memory that okay, I am a large cell, so I will keep producing the large cells throughout the lifetime. When it goes wrong, it develop the disease. So that is fine-tuned of BMR at this stage of development. And through this epigenetic modification and its propagated in a lifetime, if you have the aberration into that, so you have the disbalance and you have the disorder. But we know very little about this now, that how these epigenetic marks got implanted in a particular place. And second question is that how they are maintaining the memory type. Like since it is say one cell to mitosis, produce the daughter cells, right? So you have these marks, and then in the daughter cell in the same place of the genome that is particular DNA sequence, you have this mark, it again gets reestablished. So who is doing that? who is mediating. So that's one for my doctor. So I'm not going to teach it will be very hard for you to understand, but I very simply the some of the it's very simplistic way. So now I come into that. So in our genome, so we have two copies, right? We get from one for paternal and one for maternal. Right? And so now, as you know, in most of the cases, so both the paternal gene and the maternal gene express. So in all there are exceptions like if you have read the X chromosome inactivation. So you have in female, so you have the paternal X chromosome, you have the maternal X, but one of the X gets inactivated. So I'll come to that one. Other than that, and there's called genomic imprinting, you have read, I'm not sure. So there are also some few exceptions where only the paternal copy of the gene expressed, not the maternal, or the maternal copy expressed, not the paternal. Otherwise, in most of the cases, throughout our genome, both the copies from the father and another copy which you are getting from the mother, both are functional and both are expressive. Okay? So now, so basically now I am going to the transcription. Expressive is the you may have read the transcription. So there will be RNA polymerase, transcription factor, the promoter, the gene will be transcribed. Right? So now here what we look into that during the early development. So is there the both of the paternal and maternal of the developmental genes basically. Are they transcribing or they are so you will assume or based on the textbook you have made, please believe that both of the paternal and the maternal copy answer almost similar. So now what we found that there could be through the analysis that say this is the maternal allele, so this peak means this is transcribed. Okay. So what we found that this transcription is very different between the maternal and maternal. So they do not go hand to hand. So it is very surprising as you can see this time point. So this is a one shape at different time point. As you can see in this time point, only the maternal allele is transcribed, not the paternal. In this time point, you have both, and in this time point, only you have this paternal and like that. So what I'm taking, what is happening within the seed? So as it was thought that both the paternal and maternal allele express, so it is not the case. So it's sometimes paternal express, sometimes maternal, sometimes. Yeah. So with that, what we found, so if you come here, so what we found that you see that here this is the actual model that most of the genes, most of the paternal and maternal needs. So these are the color code you can see. So these are two allele. One is the blue is paternal, red is the maternal. But what we found that there are many cells in different time point. You have only one allele expression, sometimes both allele, and either of that. So now I come back to the previous few days, that's why I'm discussing this to the what I was discussing the epigenetic regulation, stem cell differentiation, early embryogenesis. So now we put into this actually in this pre-gastrulation is epiplast stem cells. So this kind of expression
So
of
DBT seminar.
and the developmental gene. So what we found,
Zagato.
yeah. So this is very interesting. What we see that for the developmental genes. What is developmental genes? I mean the genes which are driving the stem cell differentiation and specifying the cell types. So there are particular some series of genes which are at the forefront of doing that. So those I am finding as the developmental genes. So what we found was developmental genes, so if you take the transcription or expression of the paternal and maternal allele at this stage of development, so they actually express very much similar manner or together. So if you take the other genes which are not driving the development, they have other metabolic rules or other kind of cellular functions, but not doing the stem cell differentiation or embryogenesis. So the one very very interesting difference. So this developmental genes actually come together with a copy from paternal and the maternal So they make sure that okay, this is also fasting means the first line is the paternal one. So at the same time, maternal one also transcribe. What I explained before that many genes do not do that actually. As you see there is a difference. So when the paternal is expressing, maternal is not, or sometimes vice versa. Sometimes they are overlap. But for the genes which are driving the stem cell differentiation, so they are always of the paternal and maternal. So they are always coming together. So what now? But other things they don't care like that. So that is very, you know. So actually they work as a team. So it's a very very philosophical and interesting. So they actually work as a like a force like now, why it is important? Why it is important? So, because as I say, so at the gastrulation, so you have 30, 40 different cell types, and which are actually going to form the different immune system. And what I say, two things is very, very complex as well as interesting. They have to do very quickly. They don't have much time, and also they have to be very precise is mathematical. Why? Because they just many cell types say from the mesoderms and they fall short with the enough number of the ectoderm. So your proper development. So they have to be very calculated that okay, this much cells go to mesoderm, this much cells go to ectoderm, this much cells go to different other nucleons. Correct? So that's why there has to be butterfly beside very quantitative. And now that's why this comes. It's very interesting that how these cells do the math, how they quantify. Right? So now the developmental genes which are there are particular transcription factors which are going to be important for forming the mesoderm. And there is another transcription factor which are going to form the endoderm. So now they are expressing threshold, the level, how much the protein will be produced. Correct? So that is very, very quantitative. So now a particular cells will go to mesoderm if they achieve this much threshold of a particular transcription factor. Similarly, another will not go if they have less or some that nothing. So for that, so these join hand together both your fathers and mothers. So like this is very socially relevant, So okay, we debate like for all of us growing all the things something that mothers take care more and not the fathers. So we are society and what is happening here. So here at your genetic level or at genetic level, during your emergencies, so actually not only inherited the paternal copies and the maternal copies here, they are coming together, paternal genes and maternal genes coming together, join their hands to make our development very precise and quantitative. Okay, so opposite manner. So how you can argue that and there are many other things which are not at the similar stage, not driving the development. They are not doing like that. They are going very down. Won't see much more deep on the cell between the pattern. So basically, what I'm feeling very philosophical way. So giving this gastrulation to make the precise stem cells and the differentiation. So both of your paternal copy and maternal copy for the development of stem cell differentiation related genes. They're coming together for what? To make sure that okay, for specifying a particular cell state, certain genes need to have this much of product or expression. So that's why they come together. Otherwise, you can imagine that one paternal is only expressing, not a maternal. So you cannot expect so. So you have to come together at the same time. This cannot wait. And same thing, another same time you want to keep it off, right? So now you say paternal goes off, but the maternal will go off, you'll have a problem. So not only for actually particular expression level for proper differentiation or development, for switching off the genes, there has to be coordination from the both paternal side and the maternal side. So that they make sure okay we are going to form these things. So we are not supposed to express the other set type specified genes here. So that they make a coordination that okay both paternal also keep it off and the maternal also. So altogether, what I'm feeling that not only and then now how they can have coming back. So now how they coordinate. But it's very interesting that how they because it's a maternal zero, maternal zero. So now how they have to communicate, right? They have to talk each other, right? To make sure that okay, I am expressing now, you also express. Or to make sure I not be active now, you also do not be active. So there has to be some sort of communication. So what we found that is very interesting, again coming back to the introduction of the today's talk, the fact that how the epigenetic regulation is instrumental. So there certain epigenetic marks which actually marks the both of the addict. Addict is the pattern of like say as I said one of the mark is A483, which is actually the active mark which activate the gene expression. So now what this mark are equal amount present both lateral and lateral. And there are some transcription fact using that mark. I'm not going into all the detail, but very philosophical. So basically the epigenetic modification again coming into play makes some sort of key effort from the maternal and maternal comes of the communication with the agent. So these epigenetic marks come into play. And then you could see that where the adding the which are do not have such sort of coordination, the pattern and matter for other things, not which are developmental things. So there you also see that those marks are there. So, but it's very new things and yet to explore more that how they do that. But there is a correlation with and again these epigenetic marks. So what it was thought that they are not only just specifying the cell type and maintaining the memory that says remember their identity for the lifetime. But then instrumental, how instrumental to make the mathematical or quantitative calculation for the same cells or differentiating particularly with the RDA calculations. It might not be so calculative or mathematical in others particular states and differences, but it is very important here as you are originally from one particular cell to the potential to so many different sectors. You are forming so many different. That's why you have to very precise, calculating, mathematical, and that's why you might also new fields of learning want to be developmental bias. So actually you start learning the math, in your school, but you have started before in your stress. So a lot of calculus, a lot of things at very very quantitative level is happening when you are calculating to make sure your balance. So now I switch to another most interesting topic we also work on. So which also we will connect what I said here. So that is if you have break the X chromosome inactivation. So X chromosome inactivation means the female cells we have two X chromosomes and male you have one X, one Y. So what happens? That as male has one X and you have two X in the female, so to balance the gene expression from the X chromosome between male and female. So what happens in female, one X becomes silent. So they become inactive. So that is known as the X chromosome inactivation. So now what I'm going to tell you very interesting things about it. So that like say these are the pluripotent cells what I explained in the development course in the blastocysts collective blast. So they are actually still in the female cell of course. So both of the X chromosome is active, it's not inactivated during the early development. Okay. So now in differentiations what happens? So this two color is meaning that one X you are coming from your father, one another X chromosome you are taking from mother. Right? So, and what cells do that it's a very stochastic or random. So they choose one X chromosome as the inactive X. Some cells choose the paternal one, and other cells choose the maternal one. It's a very random thing. But now what is interesting more about this, that is what I was explaining. The likes that we say one cell specified in the early development, which this orange chromosome is got inactive. Okay. And these cells, you can see this black chromosome, not yet. This is just paternal maternal chromosome. And now the most interesting that same eggs, like whatever cells throughout the mitosis from these cells is going to form throughout the lifetime, they have to dividing, right? Throughout the lifetime, they will keep the same eggs, like here the orange is inactive, all these daughter cells and more division, more division, more division. It will remain the orange as the inactive. It is never going to switch to the other. Okay. Even there is no exception in cancer or any disease so far it has not been found that there is a switch. So now that is also it's a paradigm of what I was discussing the memory. So it is very well established today that this inactivates activates actually have very, very different epigenetic bonds. And that is also carried forward to the cells. So it activates as the inactive specific epigenetic ones, activates as the activate specific. So those ones are always that's how same to the memory. Again, I'm going back to the memory. And this is the one of the most interesting so far what I have explained and how the epigenetic memory can perform. So that's why this is a mix of the activation in my lab to study as in the modern system. If you want to know that how the epigenetic memory is propagated during development throughout the life. So because you have entire axis activated and other axis activated and memory is propagated. This is the most strongest and very interesting beautiful example of the propagation of your cellular memory. So now let's come back what I was discussing. So another very interesting things came out so from our part. Simply, so what I was talking about, like that your paternal genome and mother's genome, how they act together, makes sure you develop property at the early stage of it. So now look at what is happening in female cells. So as I said, female one is getting activity at the early stage. So what I will show here. So again, these are the early stage of embryogenesis. So at the beginning, so what happened? This is the mouse. One, both of the X chromosomes. This is two X ions indicating A means active, I means inactive. So at the beginning, both are active. Okay. So now gradually they are going to make one inactive. And what happens again? So now as soon as one X chromosome is getting inactivated, the other X, so you may not find in the text, you are only read about the X inactivation. But it was a long-standing question before that. What I'm telling you, that all of our autosomes, like beyond the six chromosomes, right? So you have two copies, correct? And now what is happening in male, you have one X chromosome, right? Female, though you have two X chromosome, one X is inactivated. And now, if you read carefully, so there are many metabolic pathways, signaling pathways. So where, so there will be a lot of organ epigenetic pathways. So there are many protein complexes formed, right? Protein complexes, there are different proteins come together. Some will have the catalytic activity, some will have the binding activity, some are both active. So all together, so not a single protein. So many proteins come together to guide the metabolic pathways or signaling pathways or any other kind of activities. Right? So now there are also many protein complex formed in our cell, like some protein which is coming from the X chromosome, some proteins coming from the autosome, say chromosome 21, right? So now chromosome 21 is producing more because they have two copies, but the X chromosome we have one copy, as one X is inactive, and male of course we have only one copy. So now if you look at the stoichiometry of the complex, so you will not able to form enough complex as you require because you have falling short for the X chromosome protein. Right? So this was long-standing question that they keeping one X active in the male, in the female, and of course male by default you have one X. So how they balance all these, and then it's very important. Genes also encoded by the X chromosome which like stem cell differentiation, metabolic pathways, signaling pathways. So it's not that the sex-related function. So there are many more important functional techniques. So this what we found. It's very exciting. Again, I'll come to the philosophically of what I started. So here also what I told you remember that paternal, maternal coming fine together to make your proper expression, fine-tune quantitative way the expression of differentiating genes here. So here also same thing is happening. So as soon as one X gets inactivated, so the other X chromosome, which is active, so they start to double their expression. So that though another X is getting inactivated, so you double their activity. So that you have almost, and then but this does not happen. Like inactivation happens most more or less all genes, maybe 2 to 5% genes which do not undergo inactivation. But this, we call this upregulation, is stopping the dosage from the active X. That do not happen for all genes, remember. But very, very important genes, which are, as I said, these protein complex forming genes, taking part in metabolic pathways. So they double their activity. So, but again, what is happening here? So, as soon as you inactivate, you upregulate, and then here will be inactivates the activation in the potent cells. Again, this upregulation got erased. Again, you get inactivity of it. So it's a very, very coordination between the paternal X and the maternal X chromosome. That one's become inactive, and others become more. The inactivation gets reactive because certain states are not going to reach it. Like here, the inactivation has to be reactivated. So as soon as inactivation again comes back to the active state, the upregulated state of the active is also come down. So see that it's a very, very ongoing, very high geometry. So now come into the another beautiful things we study, and it's a long-standing limitation of the induced pluripotent stem cell reprogram. You may have read that now we can reprogram our somatic cells as fibroblasts. We can switch them back to the what are kind of pluripotent stem cells what I discussed at the beginning. So this is termed as the, you may have read the Himalaya on the Nobel Prize for this, that you put certain kind of perception factors to say our cells. So you can bring them back to your early development back to the potentials, which has the capacity to give the all kind of needs. Right? So this we call the technology called the induced pluripotent stem cells. Okay. Right? So now what happens? So one of the obstacles of the and then there's a huge, huge impact of the induced pluripotent stem cells, not just for fundamental biology, you can switch back, but these cells can use for the regenerative medicine to regenerate part of your body and different disease modeling. So basically, the translational medical aspect is used to include stem cells has huge, huge impact. But there's some roadblock in terms of the epigenome or the things. So many times these cells when it's regenerated and you put back into your bodies or rejected or things, and sometimes develop cancer. So they're taking, so one of the major things is coming out that though these cells have changed their identity went back, if you look at the epigenome what I was discussing, the epigenetic modification, gene property set back how it was in the early development we had. So people gradually increasingly is evident that it is not. So there are a lot of fall back, fall short, that you take actual pluripotent stem cells and what you have converted from your somatic cell to the pluripotent stem cell. So from there exist all these epigenetic modifications from the genome as well as modification what is happening. So you find many differences. And then same thing, so like for human cells, so like so this is a mouse run fibroblast cells, fibroblast cells or run fibroblast cells. So you can derive from the also the fibroblast cells, so from the mouse run fibroblast cells. So in these cells, one X will be inactive, and they, as I said, the pluripotent cells you do not inactive, two X is active. So as soon as you reprogram, so then you should expect that inactive X has to be active. So it generally happen in mouse very much, but it doesn't happen in human. So they are basically for the translation approach to human interest in states. So there are huge roadblock into that for the usage of the important states, for the clinical aspect, for regenerative medicine, for the human states. But mouse it has been achieved. The culture condition, because we have to establish a proper environment. We have to recapitulate an environment like the embryo. So why these cells become embryo-like components like cells? When you achieve that, it should be absolutely healthy or very much useful for the regenerative medicine or clinical aspect or disease monitoring. But mouse, I come back to the mouse. That's the root of what I was discussing. Mouse also is there. But Another interesting thing is happening. So in this embryonic fibroblasts, one X is inactive, another X is upregulated. As soon as you convert to iPSC as this inactivates X gets deactivated. Correct? And similarly, so this upregulated X comes to down, we use that expression. So that now both of the X chromosomes has almost equal expression. So this is happens very collaborating way. And as soon as this goes little up, this goes down, this goes up, down, again this goes up, this comes down. So we are very happens not only in the early development in CSA, it will first back development in the English steps. So again go back like that. But again, as I say, human it's not happening like because what I can think that in human the protocol of the technology used for the program will be used for the mouse, but there are a lot of basic difference, though there are many things are constant, but gradually you are learning there are huge differences. So mouse development to the human development. Many things are same almost. But there are gradually you are learning there are many differences as well. That's why what works for the mouse IPSC program works also for human, but it does not work as efficiently. Efficiently not only comes to numbers, like the quality of the IPSC program. That is their epigenome has been reset appropriately, what it should be a plant stem cell, or like you have to put stem cell in your so there is goes back or gap. So that's fundamental law. And what is this topic is the skills and technology and biology. So I think that's what it highlight from my lab was from basic fundamental biology. So there's a huge roadblock. I think that has to be crossed. And the roadblock is running basically many things are getting reset, but you are unable to appropriate reset in teaching of different marks, how you have to reset their human. So yeah, so just to end what I'll touch now, it is human likeness, so we did some work and how we can improve the human potent stem cells culture, quality, all the things technology, so that we can gradually reach. So we make a human potent stem cell lines. So where you have the appropriately all the things, so that you increase that it's really useful for regenerative medicine. It can be successfully used for disease model without any limitations. So to we started with the X chromosome. So as I said, the including program. So the X chromosome state not get established like in mouse. And same thing. So as the X chromosome state has been understood in case of mouse, in early development is the say type to 8 cell state, plastina, all those things. In human, we've been able to study much because mouse we can break the mouse, we can isolate the embryo, but for human is the ethical and technical challenges. So for the human, it's not easy that you go take the embryos and do different. You have to do a lot of in-depth molecular level studies, experimentation to understand all of these. That is remain obscure. So that's why it is assumed that human are the development epigenomic things or the X chromosome inactivation is most likely the mouse. So now when this in vitro fertilization technique, so a lot of infertility thing even in India, a lot of infertility issues going to be solved with the in vitro fertilization. So there you can get some of the human embryo where they do not use for the injection for their making their baby. So because they make a lot of in vitro fertilized embryo and they do a lot of quality check and then which are in terms of very good quality they use, which are bad they do not they just freeze it. So we use those embryos because we don't have other alternative to study that okay, first we started with that what is happening in the X chromosome state, which is similar to the mouse or not. So there we found very fundamental difference between mouse and but again there is a limitation as I say that we do not know because the embryo we are choosing is a little bit abnormal. It's not such a high quality. Okay. So we do not know what happened. and this kind of study has been done in other lab also. So they have done some sort of differences. But so far we get consensus from different group of studies that it is absolutely not happening like the mouse, that at the beginning it is inactivated and then it's remain active for very long time, and maybe after implantation it will start inactive. So I'm not going to detail. So what I'm trying to tell that now human equals from home. Now I'll not claim that we know complete or what we know is completely correct, but definitely from my lab or from some other lab of Europe and America, so we have some sort of progress both on the extrovert state and the indigenous state. But that's not the thing. We have to travel more. And that act as a reference also. That human IPSC, what should be the ideal state? We do not have a reference. For mouse, we know that early embryo state should be epigenome or whatever state. That it should be like this, there, correct? But for human, you do not have reference. So human IPSC, so what we are comparing, we don't have a true reference to compare. So this is the roadblock again, because the studying the RD embryogenesis in human is still very difficult and very there is a huge ethical concern, correct? But now I'll just say that so some of the translational aspect from this knowledge what my lab is trying to solve. Okay. So there, as I discussed, in vitro fertilization clinic, we have the technology. So basically, fertility problems, you can take the sperm and site from here, and sometimes you use donor, and then you fertilize them in a cell culture model or tissue. So outside of the body, so very artificial. So it's very very sophisticated. Right? So now, what one concern, as I said, is in vitro fertilization and quality control. Okay? So now there are huge debate on all the things, propaganda, some people say it's propaganda, but there's a scientific evidence that though we are getting for quality control, human embryos and implanting these babies, but there are chances of developing cancer and data is much more, but it's debated. I'm not going into detail. But one aspect is that we need more quality. And another very interesting aspect, which is extremely relevant to our socio-economic condition of our country. So what has been found that worldwide, this is not India, so the data has been statistics, so so this in vitro fertilization conceived infants, so always the babies born, so this gives us more number of male baby borns. So females are always less. Okay? So as you can see that it has been worldwide estimate that if you do the in vitro fertilization concept infants, you get 56% male, and there's a huge difference. The female is 44%. So there's almost 10-12% difference in the gender in case of infant born from the in vitro fertilization. So now this might not be so scary for the other countries, but if you look at our socio-economic condition of India, so there already we have already damaged, So we have the mentality in our society that we need son, not the daughters, right? So yeah, so that's why we have huge sex ratio towards men. So we have more men than women today's. Okay. So now, if you see, if you go to the Bangalore or Hyderabad or some major city, you find so many fertilization clinics today. If you go 10, 15 years back, it was so that's what they are doing good business, and that's what it is indicating that many patients or people opting for the in fertilizers. Right? So now, and then so we have already skewed sex ratio, and many people opting for the in vitro fertilizers, right? And that will also again induce, and then there are corruptions also. I'm not going into those are very, uh, political and scenario. So I'm not going to that. So, but in normally also, so if we are gradually opting more and more in vitro fertilization, and also infertility rate is also increasing. Correct? So many countries infertility increasing, and obviously our economy is improving. So many people are going to opting for the fertilizers. If that is the scenario, So we are going to create more and more an imbalance in the sex ratio, and which is already we have to try to reverse. We need more women. And we are going towards this, so this is going to have that huge basic confidence. So now how we can solve it. So now I come to the science what I discussed on a couple of last slide, that why you might be curious that why when you are doing in vitro fertilizers not through normal procedures, why you are getting more male, not female. Right? So that has been from my work and other works, other groups' works. So it is now what we found that major difference in the male female is that female skin activations are the male not activated. So it could be we hypothesize that this in vitro fertilization in case of female, their X chromosome inactivation or what should be the actual X chromosome state when you are doing artificially, it's not very easy. And as I said, the gene expression from the beginning to what I'm trying to express from the, it's always important. For the RT development, it is important, not only very quantitatively important. So we have to fine tune the expression. So now your X chromosome has very large number of proteins which encode from the X chromosome and which directly take part in the stem cell formation, early lineage formation, blastocyst, the different lineage of the blastocyst, ectoderm, mesoderm, endoderm, pluripotent differences. So they are coming from a lot of genes from the X chromosome. And if their expression is imbalanced due to the inability or inefficiency of the dosage compensation of X activation, so that creates that embryos development get fertile or huge. That's why how many male embryo you plant and you get the successful implant. So many times female you do not get successfully because some of the embryo have defect of this X chromosome state or X chromosome deficiency. So as I said that obviously, so now we have made a progress. Now what we have a reference that how an embryo, a human embryo, which is going to be implanted, what should be the X chromosomes. So with that reference, what we are trying to do that if we can, you know, rescue that X chromosome defect. So that's technology we found, or at least quality control that. You know, like say you pick 100 embryos and check that which actually reflecting the proper X chromosomes. You only choose. So far we did do that, and which do not have so discontinuous. And then when you do that quality control, so you do not lose any implanted abilities. And so that what this ratio is going down in this of female because a lot of females placed in their market because they're starting to develop the market. So that can be repaired and then of course we do that practice possible. So then you establish the proper male to woman sex chromosome ratio. Sorry, sex ratio. At least in the babies born to in vitro fertilization. So this is we have a big hope. We are working very excitedly into this, and it has very much national importance. As I said, I laid up here. So we need both daughters and son, and as I said, the whole philosophy of this talk beyond the science. So father and mother, both of the six is very much equally important. And we are talking about the women empowerment, so there it comes. So we need empowerment even the in vitro fertilization, as you can see. So we have societal defect that already we have skewed sex ratio towards more men than the women, and this is added another problem. So we have to solve this. And so basically, what I'm going to aim that both of the sex, in terms of philosophy, you go to the epigenetics, emergence, stems, frequencies, and the, your development. So there, both sex is important and equally important your other contribution and other contribution. And we calculate the quantity. So with this I'll end up and so this is my some of the work I have said here. It's funded by the one of the very popular European molecular biology organizations. So I'm a member of that, and of course Department of Biotechnology, DBT Government of India, CRT Government of India, of course, a lot of help from Indian Institute of Science, and also support from the Infosys as well as from the MAR. And some of the work I described today, these are the three graduates today who contributed very much, very significantly. And this is my group. And thank you very much for your attention. I hope I'm not sure how much, because as I said at the beginning, so this is, I'm doing first time this kind of lecture, so maybe I'm not. So if you have a problem, feel free to ask the question, I'm happy to address, or whenever after the presentation, some late or something. But I hope I have excited you that again, online, that how the epigenetic regulation during the early development stem cell differences is important. And understanding of those fundamental things, how is important to solve the challenges in the iPSC technology for human, both the clinical level and also to more directly to solve this X-ratio problem of the immunopathology. And then top of that, that how the both male and women is equally important to our not only our society but for our development, our embryogenesis, stem cell specification, and of course, and father and mother work together to drive your research. So thank you very much for your attention. Thank you again, Tony, for this song. I really love this to present such kind of work. Thank you. I hope I finished it. Yeah, little. Any question from any of you? Feel free, That's a very technical, you are curious something, don't hesitate. Yeah.
Ramakrishna, come here. Introduce yourself and then ask your doubt.
Good morning, sir. I'm Seth Ramakrishna from Government.
Yes.
My medical. So my question is, the placenta is using as a pluripotent cells. So how is actually useful for the further? Actually, they're storing for cryopreservation. What is the need for that placenta?
Yeah. Not only the placenta, other stem cell preservation is coming, and in our country, it will be as well, going to be popular like today's fertilization as it is. So, yeah, so same thing, if you can preserve those early stem cells, or so when you need, like what is your or what different other purpose also. So you can take those cells and you can see specify the limits what you want. Of course, that technology has to improve it all. But there's a huge hope and we will already start. So that you utilize instead of converting to the IPSC by the limitation, you take directly from those cells and generate what you want. Okay? Okay. Yes.
Sir, in my observation,
Yeah.
in IVF centers,
Yeah.
mostly boy child
Yeah,
born.
exactly.
Is it intentional or accidental?
No, that's what I'm telling. I think going through that. But let's see. I would say so. This statistic what I present, taking average global, not only India. Now, if I take India, I don't want to present that. Okay, so it is hugely biased. And then I believe so. Now, if you compare the say the data from Australia, Europe, or America, so there are bias, but not like India. Okay. So of course, in general, I think that's not only for the scientific problems, there's a malpractice. And because of that, but to go like this kind of malpractice or there's no societal emotion like boys versus girls in other countries. So they are also will find this issue. So that is definitely compounded with by malpractice. It's real. So it is real that there are some of the female embryo because of the improper exposures. Because we do not know what at the improper exposure state of the human embryos. So we never call also. And female because of this exposure inactivation response they happen. So you are implanting the embryo which are not proper. And that's their type. Male you do not need to do all those, so male sometimes. Male also type, there are other quality of that. But when you directly compare male female with the same quality you are implanting, males are more surviving. That data is absolutely true. But if I present the only Indian data, it's very hard to predict how much is coming from. And that's why it is more ratio bias is there. gap also. Yeah, and I believe there are some that. So I don't want to go there, but take the global data, so it is there. So at least if we can resolve that problem, it will definitely bring some improvement. And this malpractice, this is I think proper scientists job and social worker and political workers, and all together we have to be more educated. I think it's changing also. The good hope is there. We are changing.
So in India, sex determination is a crime, but opting a sex is not a crime.
Yeah, 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 absolutely.
Thank you, sir.
Yeah.
Dominance of the Y,
Dominance of
the Y chromosome.
yeah. Yes. Yeah. Could be. Yeah.
Whether in vitro and in vivo,
Yeah.
still the Y chromosome
Yeah.
stands dominant.
Yeah. Yes. Yes.
Sir, I have a doubt actually. There is one IVF center, so many IVF centers here.
Yeah.
But most of the cases through IVF, when you got resultant baby, most of the cases the boy sex. Is there any possibility to select X or Y from by in vitro fertilization?
Oh no, that is you can do all those, but that is strictly prohibited that you don't do
No,
all.
sir. It is possible, sir. It is possible.
Yeah.
Is it possible to either sperm contain either X chromosome or Y chromosome to detect? Is it possible or not?
Yeah, it can be. It's possible.
Okay.
Yeah, it's possible. All the things is possible. Yeah. Yes.
Sir, if I may add to that, because I did a project on that, we have a technique which is known as ICSI,
Yeah.
intracytoplasmic sperm injection,
Yes,
wherein we
yes.
can select the sperm, a single sperm is selected.
Exactly. Exactly.
And then we have the choice, yeah.
Yes. Yes. Absolutely.
X or Y
That's,
we can
that's correct.
that be a part of IVF.
Yes. Yes.
So, do we have any more queries from our participants?
Yeah. Yeah.
So, we have students who have come all the way from Women's College. Anybody who wants to talk anything? Language is not a barrier, we can translate for you.
Yeah. Yeah. Absolutely. Don't. Okay. All right. I think you feel free even after that.
Yes, thank you, sir. So we thank sir for coming all the way from Bangalore and interacting with our students. And I hope our students have been motivated. And we have participants in online too.
One question like.
So we have a question from Mr. Arun. Sir, induced pluripotent stem cells (iPSC) can be used in treatment. Is there any chance of getting uncontrolled proliferation due to mutations?
Uncontrolled proliferation due to.
Yeah, mutations when using iPSCs.
Yeah, no, that's what I was stating. That though this technology we have decades and decades, but still there is a huge roadblock. So this is one of the aspects that there can have some mutation because you are converting the cells in the same culture, and there are a lot of aberration, the genomic changes is happening, like the mutation, loss of chromosome, deletion of a particular part of chromosome. These are very well reported. But still we can, you know, quality control, you can sequence the genome or the thing and then choose some iPSC lines. People are doing that, which has been progress very well. But yeah, of course, if you take those kind of iPSC and you apply for regenerative medicine, I think it's not block, it's not going to be successful because it causes some form of different kind of problem and it's not getting also implanted properly. And top of that, what I emphasize, what field I'm researching, that we only talk about the mutation or the things, but the genome is much more important. So like if I install modification, DNA methylation, all. So they are also to compare, so it is not up to the law. So, but it's not easy to restore those. And rather we have to improve the technology of protocol so that when you are generating the IPS, you improve those. So if it so it definitely a lot of research is going on and the papers you it will improve. And then definitely it will be good.
So I think Arun sir has answered your questions. Yeah, and sir has responded as thank you, sir. I think
Yeah.
his query has been
Welcome.
clear.
Yeah.
So In case, do we have any more queries from online participants? Right, sir. I think we're good. On behalf of all the participants and the organizers, I thank sir for spending his valuable time and interacting with us. And sir will be available here today, students, so you can interact with him on personal basis also in case you are feeling shy to ask in front of everyone. And we come to the end of session one with this, and we'll take a break of 10 minutes. After that, we'll reconvene for the inaugural session. Thank you all. Students, I request you to remain in your seats. Snacks will be distributed at your seats, please. Please remain in your seats. Thank you. Dear participants, can you please assemble back in your seats? I request all the participants who are outside to please come back and be seated in your seats. Students, we have chairs here. Please come forward. Settle down fast. We are moving on to the next session. Dear participants, we are now moving on to the next technical session, which will be headed by Professor E. Vijayan sir, currently the President, Society for Biotechnologists India. And sir has been the recipient of several awards, including Dr. Subhash Mukherjee Memorial, Infar India Oration Award, Psychological Society of India Oration Award, President Endocrine Society of India, Council Member International Neuroendocrine Federation, Emeritus Member Endocrine Society USA, and served on the editorial boards of several journals. He is widely traveled across the world, participated as invited speaker in numerous national and international conferences and symposia. On superannuation in 2006, Dr. Vijayan sir was selected as ICMR Emeritus Medical Scientist and joined the Department of Biotechnology, CUSAT, and continued as UGC Visiting Professor. Sir is settled in Kochi and he keeps very active and frequent visitor to Europe, USA, South Asian countries as invitee to various scientific meetings and conferences. So we are very fortunate to have such a renowned scientist and professor with us. And I think it is a privilege, sir, to hear you address our students and ourselves. So I hand over the session to Vijayan sir now. Thank you, sir.
Thank you all. Thank you for the nice introduction. It's a pleasure to visit West Bengal and this college and be here. And Dr. Pradeep is so proactive, and it is because of him some of us come here again and again. And you all should be fortunate to listen to whoever is. We had a beautiful lecture in the morning on stem cells and how the in vitro fertilization and the birth of male female children can be. There was a question, interesting question answer. So it is for you to solve that puzzle or problem of how to suppress the dominance of Y chromosome, and you can have both male and female children equally, or you cannot have equally, but this male dominance should be abolished or brought down. Okay, so you must take lead in that. That's what all these meetings and lectures will help you to understand, and you don't know which way your future will take place. Thank you. So I'm glad to speak this morning. On something in general. I haven't done anything on stem cells directly, so I'm not a stem cell biologist, but we have done several things. Whether you use cell lines, you use cells, and you study how communication between cells leads to what you heard in the morning, what you call the development. Okay. So I'm going to give you a few ideas. These are not directly my work. No, this is just to motivate you. A few years ago, 5, 6 years ago, I happened to attend a meeting in Geneva. That was in connection with the every year all the world's economists, famous economists, and the head of governments meet. It's called World Economic Development Forum. Okay, and so that is not heads of governments comes. They are not, of course, they happen. So many of them happen to politicians, but they have nothing much to contribute. But they are because economical aspects they give. So this they have scientific experts. So this economic development forum invite world-renowned scientists, and they have entrusted the scientists to formulate what are the uh, scientific development achievements and what science has in store for the future. Okay, so it is a scientist could identify those areas which is already being uh, explored and researched by several people in different parts of the world. So some results are on the way, some are already. So these were the discussion at that time, 6 years ago. Okay, so I was since I had a chance to attend that meeting, I felt it was so impressive, and some of the theme thoughts which I gathered from that at that time they called disruptive technologies. Okay, now there is a forum of scientists selected by Scientific American. I don't know how many of you know. There's a science magazine called Scientific American. If you have a chance to read, you should read it. Still, it is published every month. So, this group of scientists identified certain areas which is going to change the world. So, you can imagine the importance and significance that word itself. The developments in science which is going to change the world in the next 10 to 15 years. So that was the theme somewhere 2015 or 2016, I don't remember, more than 5 6 years. Okay, 2016. So at that time, all these discoveries of the development in science, it was in such a stage they could uh, propose that these ideas, 10 or 15 ideas which they identified, will change the world in the next 10 to 15 years. That means by 2025. Okay? And now, when I give you a lecture now, after that 8 years, you are already, those technologies I will show you, already on the on application or being applied. So, many of them, and that changes are being seen and felt. Okay, so I thought that being a few technologies which I got from, I got this idea and then from those things. Okay, so nothing my contribution, but the ideas which are contributed by several leading scientists in the world. So I'll show you about 10 of them, which is that's why they called it at that time disruptive technologies. Now what is you know what is why they called it disruptive technologies? Anyone knows what is disruptive? Not destroying technologies. Disruptive technologies they call it. Which is the disruptive technologies, which is going to change the world. That was the theme, focal theme, heading given by Scientific American. Okay. Now, I also felt what are the technologies, disruptive technologies, which is going to change the world. You know, it's like atom bomb or hydrogen bomb, which kind of force destroy the world and change that's immediate, but it is not. Now, anything disruptive, why they call disruptive technologies? These technologies are going to replace the existing technologies. So these are new technologies which will replace existing technologies. That's why they call it, it will, when you replace the existing technologies, it will change the world, face of the world. Okay, we are right. So I'll just show you a few of them. So the middle one, you, you change the middle one or? Okay. Okay. See them, they call the emerging technologies that are poised to change the world. Okay? Now, disruptive technologies are innovations that significantly alter the way consumers, industries, or business operate. So that is the replaces existing systems and or habits. I already mentioned that I don't know that's one already World Economic Forum's experts work in collaboration with the scientific community. I already explained to you, so I don't want to repeat and take time. Now, example, for example, what if drinking water could be drawn from atmosphere without requiring any energy? You know, the drinking water, I've been sitting and drinking water. Sensors all over the world, the water is now many places, you know, to clean drinking water still and should in many places will be getting. So in such a situation, you take a tumbler and stand out, throw the tumbler, and if you can collect pure water in the tumbler and have it, water, isn't it? That's where they call it disruptive technology. I already thought we can do that. So scientists have been working on that since last 30 years, and by 2020 or before that, they have produced the ways and methods so that you can take a tumbler, stand out anywhere, any part of the world, in the desert or Stockholm city or anywhere else. You stand for 2 minutes, you can collect water in your tumbler. And what you collect will be clean water. So what a wonderful idea, isn't it? Whoever felt that idea, that's anyway, many scientists were involved, they start working on that, that become a reality. What is now going on? Already they have several parts of the world. They have experimental fields or places where the amount of water collected, desert region, semi-arid region, arid region, or dry regions, or it's there. And what is now the most important, you must have the number, not the original number. So the chemical, that is why the chemist and physicist and biologist involved, they have developed the chemical basis by the which so the manufacturing of those vessels and utensils are now the current technology. Scientists have already provided the scientific methodology and scientific basis. Now it is like now it is the manufacturers which are taken over that is so you can have the commercially anywhere in the world. Those vessels are available. You can buy the vessel, and that vessel are cheaper. So it is one of the exciting ideas. Okay. So that is, then second part is the doctor will do biopsy without a blade of metal. So biopsy is a medication that is invasive procedure. So the doctor can could do biopsy without a blade or that would be that's also. And then what if we don't have to wait too long for results. Earlier, my days, sensitive assays, something bioassays, anything if you do a bioassay, you have to take 4 days. Then by 7 days, you want sensitive radio analysis. Then you have results from the microbiology students, molecular biotechnology, molecular biologists, all the techniques now available. Okay, within now time you can get the results. Then diagnosis and that's the next step. So like this, we don't have to wait too long. The technologies make these visions a reality are expected to become commonplace in the coming years. So there will be just just an example I give you. So we have several technologies are on the way, which is replacing the old technologies, and they are going to use that's what is going to they call change the world. Okay, that's only the so we have few more years now. We are already so by 2030, but few technologies have already replaced in the large where okay now water made by itself. Now the technologies that pull moisture from the air without electricity. That's a wonderful idea. Okay, extracting water directly from air will be boon for billions of people. We mentioned that the systems in development rely on energy from sun. That's what is happening now. What they call the development of crystals called metal organic frameworks. MOFs absorbs moisture. So that the chemists have developed this substance which they call metal organic frameworks. Okay, that material has the potential to absorb moisture from the water. So this is the basic idea stuck to the scientists who are doing that. But then they developed on that. That now they have to incorporate into the processor, then collect water from the moisture from water and water. So this is the basic scientific principle of it. So you all should interact. Only if you interact among yourself, discuss science among yourselves, that will stimulate your brain. And that will lead to new thinking. So you should have creative thinking. Okay. Always that will lead to new ideas and what you call innovations. And present day without repeating the same old systems, you cannot survive. You have to come out with new and creative innovations. So now 1 gram of MOF, the size of a sugar cube, has initial internal surface area equal to that of a football field. So it is such a small 1 gram of a sugar. That size has size surface area of that. So that much surface area will absorb moisture. That's the principle they got. Now, the device can harvest the coming device, what is available, 2.8 liters of water per kilogram of alcohol at relatively low levels of humidity, as with low 20% levels of humidity. Uh, Based on that, startup called Zero Mass Water has begun selling solar-based system which can produce 2 to 2.5 liters per day. Then you can add calcium and magnesium can be added for next. So that's the technology which is now available. Now when I made this, it was 2.5 liter. Now it's small. They have storage device mechanics where it can up to 50 liters. You can correct something. Or what you have to do is the calcium and nothing is looked from pure air as well. Unlike today's river water, who correctly go through the process, because we dump everything in the river. So you can't use the river water. So, so that's following. Then next, fuel from artificial leaves. We all, as students, we learn how fuel is made from fall as a result of photosynthesis. Okay? So technology that mimics photosynthesis to generate fuel that could be stored for later use. Like a real leaf, the system would use only carbon dioxide, water, and sunlight to produce fuels. Sunlight activated catalyst split water molecules to yield oxygen and hydrogen, and creation of a closed system in which CO2 emitted by combustion transformed back into fuel instead of adding to the greenhouse gas effect. So these are all the technologies to create. A plant uses just 1% of energy from sun, whereas artificial system achieve 10% efficiency in converting CO2 to fuel. So that's also, this all 10 years back, they predicted as a result of 20 years or 20 or 30 years prior one. And so today it has become you hear about the cars, you get the fuel efficient cars, so many of them. So we have already started changing the changes. Artificial intelligence that sees like human. Every day now you open the paper, you hear about artificial intelligence because AI-based systems they started misuse also. That's why it has started coming appearing in the newspaper. But practically now we have everything based on artificial intelligence. Okay, that's so they predicted the long back in the system. Now, what is called the deep learning tool for virtual task changing medicine security. Breakthroughs in deep learning enable computers to interpret images successfully as or even better than people do from driving interpreting scans for medical diagnosis. Now you must be reading about cars or the cars which stop at the zebra crossing, vehicle automatically stops. All this is because of the artificial intelligence based what this based what you hear about it. And so what is it? Everywhere we use that. Here we use when I press it changes because it has something. What is that? Can anyone say? Artificial intelligence based. You put your clothes in washing machine and you just program it. There are different programs for 5 minutes or 10 minutes or stain removal or light washing or 5 liter water use or 15 liter water use for everything is programmed. But what is that? Your automobile now we use. No? Reading is a must. Okay? Sensors. Practically everything is now based on our life is now going to be used. Already we are now in the days ahead we are going to the artificial intelligence makes sensors are going to control our life or we can control it. Okay? So that's all very good. Now so that's computer vision system now the scanning you hear about scanning. The scan is done, the machine itself will read the scan, then write the report. The doctor only has to read the report. MRI or that scan or this scan, you have so many scans. But it has also has the earlier the doctor has to look at the X-ray against light or against sun, then uses imaginative, creative thinking, then diagnose. Today the machine diagnose and give you the, then the doctor gives you the what is to be done. So that is the level of life has been changed. Now, in agriculture, deep learning technology is used to predict crop yield, monitor water levels, detect crop disease before they spread. So these things are how important you can see. You can using these sensors, they can predict in ahead. So you can take preventive action or preventive steps. Can seat experts' interpretation of scans and pathologies rights provide artificial information in places that lack that's the learned people refuse to go to villages. But then, even in the absence of the personnel there, if you establish the machine, they can read the scans and then provide the report to the and so one can visualize the blood flow in the heart, diabetic retinopathy, oxygen, all these are done by machine. Precision farm mentioned that sensors, imaging and real-time data analytics improve farm outputs and reduce waste. All these are waste. Tremendous inputs in agriculture and they have already started using. Now, what is the one which is now the police use, farmers use, engineers use, flying around and photograph journalists use? What is that? Eh? Drones, yeah, that's it. See, so in agriculture also drones are being even in India. Now, farms agriculture drones are being used in the initial stage. The drones have the sensors they can detect what water level is there, how much is deficient, how much is more, which plant is disease. Instead of earlier you put the insecticide for the whole field, now the drone can sensor can pinpoint how many plants or which area of the plant is infected. So instead of putting insecticide or medicine for the whole area, you can pinpoint the area and the plant and the part of the plant and then apply it. So the farmers save money, time, and input. Okay. So because of all that is the basis. Sensors and camera equipped drones send messages and data on individual plants, stem size, leaf size, soil moisture to computer, and the farmers receive the feedback and help farmers when to plant, harvest crop, can improve time management, reduce water, chemical use, produce healthier crops, and higher yields. So all these things are now being available and being used, including in our own country. Okay. That's why they again call so these technologies are replacing the old predictions, old experience. Those predictions are made from your number of years of experience. Like our India, the weather forecast in India has changed a lot. 20 years ago, we always make fun the broadcast from radio or TV that rain may come or may not come. That used to be our prediction. Okay. But today things have changed. At least to some extent we know now when the cyclone comes, when the air comes, when the humidity comes, when the air comes, when the temperature goes to 48 degrees like that. So that's because of the technology advance. But still in that area we have to go a long way. Now, mapping every human cell. This is a project which is your human genome project. It's completed. Now, this is another project which is now going on. Now, to deeply understand how human body works and diseases arise, how to identify every cell type in every tissue. Now, what do we do? We identify an area or a tissue or a gland or an organ. Okay? But the entire organ is not affected. The entire cell population in that tissue or organ is not affected. But now we are detecting that way and we are treating that. So you give existing medicine also, which is based also. Okay, in terms of now this project is to avoid that. Which is still now exactly which genes, proteins are used so that have the lecture where you can X chromosome, Y chromosome, and on genes and after it. So our molecule active and one process control that activity, how cells interact with one another, and what happens to body's functioning when genetic or other changes occur. Building such a rich, complex knowledge base seems impossible, yet an international consortium of scientists has taken steps to create the human cell. They call it human cell atlas. Now it's a long-term project. It is going on. International consortium of scientists. The advantage is when you can see the details of every individual cells in the body, which is a major task. But if you are thyroid gland, or if you have tissue pancreas, at least that particular tissue, you know. If you have the cell details, you know how much. So this is the way it should. You can treatment only to pancreas or thyroid or gonads or spleen or intestine or whatever specific areas. And you know the cell details, the architectures of individual cells. You can target the drug molecule to that particular cell or that particular cell group or the site. So you save on every front. So that's the now the successful combined information of the existing and future results made feasible by a host of technological achievements which include isolating and profiling proteins in single cell, quickly sequencing and all the procedures. Integrated results lead to a tool that will stimulate all types and states of the cells in our body and provide new understanding of disease processes and ways to intervene for improving our personal health. So these are the future predictions, but several things are already on the day that you know. Liquid biopsies. I'm telling you about the liquid biopsies. Now we have instead of 2 ml, 4 ml, or 10 ml of liquid blood or other serum or liquid, we can have a drop of blood, a drop of serum. And from that drop, the sensors can analyze and report to you the ultrasensitive tests promise to improve diagnosis and care. Biopsies are invasive, I told you, and cause infections and other complications. Liquid biopsies find signs of cancer in a single drop of blood sample and promises to solve many problems, amplify and sequence DNA rapidly and inexpensively. Just to know to read out now. Uh, detect full spectrum of mutations. Liquid biopsies may one day provide a fast, easy screening test for detecting cancer, its type in people who seem perfectly healthy otherwise. So that's why the cancer can start at any stage, but it appears when you detect it, it's already late. That's why most of the cancer patients do not survive. Okay. Yeah. So that's the advantage now. You can hydrogen cars for the masses. Now this is under electric cars, hydrogen cars. They also, now past 2 3 years, the number of electric cars on the road, you will see everywhere you can see electric cars on the road. Now you can imagine 10 years from now. It's not the case. Meanwhile, we have the hydrogen fuel powered cars are also on the So this is the goal of sustainable transport system demands zero carbon emissions during driving and also during production and distribution of fuel. The other challenge still remains because genomic vaccines. This is also those who read the papers or magazines, you know about genomic vaccine. We have this introduction. The promise to offer many advantages, faster manufacture and virus etc. Corona. You see how because of the technology, how we have successfully managed the COVID in the last 3 years. Because we could millions and millions of doses of vaccine produced because the technology was available. scientific manpower was available, so we didn't depend on others. So it is a thing. Producing genomic vaccines is less expensive and a single. But there is a long way to. There are some other things on the streets. If you started, there is no control. It means every second the car will stop. Because the car won't. So that's what these are the other added activities, facilities, which was created. Our behavior has changed. So these are often associated with successful etc. But the most powerful idea is the bring out of this thing, what? Keeping what? That's a reality now. Many places they have already going to start, especially it's coming to India also. They have experimental studies to do that. Is not many of the these countries started doing this as well. Now, what they want to increase the capture of increase the quantum of market innovation and reduce the price of. So usually that's another reason this parts manufacturing of these things they will try to make the maximum profit and that has to be factored. So they will make this so future cost is successful. There is no so what I want to tell you is think of this kind of ideas. Okay? You must invite what is good and creative, develop creative thinking. Discuss some of you will succeed, many of you will go a long way to do things. So not everybody will be successful. Definitely new ideas will come, but then we have to interact with other situations. Okay? I will show you the last slide. So anybody wants to ask anything? Anyone want to ask anything? See, all these technologies, none of these are my contribution. Again, my area of work was different. But I told you I got motivated to read and know more about this because I listened to speakers presenting this and how wonderful these ideas. So, if you have any questions, please. Nobody has any. It's okay. No problem. You're all hungry. Can any of you suggest any other technology which will change our way of living in the next 10 years or 20 years? Or you want to go with the same what you are enjoying now? See, 15 years ago, nobody talked about electric cars and hydrogen cars. Isn't it? Because of this. Anybody else? Not at all. Okay, thank you for listening since you know that. Anyway, I am not expert and I told you in the first it's not that I haven't contributed anything in that area other than reading. Okay? Reading and listening.
Thank you, sir. I think that is more than enough that you have been inspiring our students. I hope they have been inspired. So, do we have any queries from online participants? I don't think we have any queries from online participants also. So, we thank Vijayan sir. Sir is a regular visitor to our college. He is in fact our benefactor, our mentor. So he's been there with us since I think 6, 7 years, and he's been mentoring all the students. Every batch of students have at least listened to one seminar by sir, and I hope this trend continues, sir. And I hope you'll be there with us for many years to come. And We'll be proceeding with the inaugural session in a few minutes, so I request all the participants to please remain seated. And after the inaugural, we'll be heading for lunch. Lunch will be served in the Silver Jubilee Auditorium. And post lunch afternoon session will be in online mode since this is a hybrid conference. So we'll be sharing the link and I request all the participants to please join in the link. You can come here and join or you can join at the place, your convenient place. But tomorrow morning, I request you all to reassemble in this hall since morning session is offline session and afternoons we have online sessions. Thank you for your patience. Good afternoon, ma'am. Good afternoon, ma'am. Our principal ma'am has joined us. Ma'am, with your permission, we can proceed with the inaugural. A very good morning to and beloved President and beloved Dr. P. Distinguished Professor E. Respected faculty and dear students. Welcome to the National Conference on Emergent Stem Cell Technology in Biomedical organized by the Center for Applied Sciences and Department of Geology, Government College, Management, at the very audience, our President Dr. P. Onto the dais. Mrs. Raksha will welcome. It's my privilege to invite Professor E. Onto the dais. I request special invitee Sir Sriram sir onto the dais. Miss Vanita, welcome sir. Now I welcome our Vice Principal sir, MPA Satyanarayan sir onto the dais. Chandra, welcome sir. I now call upon our activity coordinator, Dr. R. Helga onto the dais. Miss Kalyani, welcome. It's my pleasure to welcome the head of the Geology Department, Captain Dr. Y. Purnayu sir onto the dais. Miss Vanita, welcome sir.
warmly welcome to the program coordinator and head of the Center of Applied Sciences, Sri and Pradeep sir, onto the dais. I request all the gatherings to please stand for the prayer song by Kalyan. या कुन्देन्दु तुषारहार कमला या शुभ्र वस्त्रान्विता या वीणा वरदण्डमण्डितकरा या श्वेत पद्मासना या ब्रह्माच्युत शङ्कर प्रभृतिभिः देवै सदा पूजिता सा मां पातु सरस्वती भगवती विश्वेशजाड्यापहा I now hand over the proceedings to our principal ma'am.
I request all the dignitaries to please proceed on to the lighting of the lamp. And I request the participants to please stand up, please. Thank you.
Good afternoon, everyone. A hearty welcome to Professor E. Vizian sir, President Society for Biotechnologists India, respected dignitaries and dear students, resource persons, and all participants. As the President of the National Level Conference, that is offline and online hybrid mode, on emergency of stem cell technology in biotechnological field, biomedical field. I have to honor as welcome to all of on behalf of Government Degree College Men's Srikakulam, student and staff. It is a great pleasure to me to declare open national conference and welcome to all the participants from the all over the nation. Thank you for giving this opportunity to the DBT and SERP. So both organizations, national organizations. So overall the nations including all participants including Tamil Nadu, Kerala, Karnataka, Telangana, Maharashtra and most of the scientists and the youngest scientists are also participating in this seminar conference. So thank you so much Vijayan sir and Sri Mohan sir. Sorry for the inconvenience. So welcome to Srikakulam again. We already met before so one successful program, but again we gathered and conducted this program. So I extend sincere thanks to two institutions, so that is DBT New Delhi and SERP Research Board in India. So we have these two are sponsored for this program. So thank you so much. I wish the workshop all success and all the students please utilize this opportunity. And in the offline online session, most of the people participating in the online. So they are also welcome to all the online participants. So please utilize this maximum time. So I welcome to all and wish you all success for these 2 days. Thank you for giving this opportunity. And so I would like to invite this program coordinator, Dr. R. Harita. Please give your words now. Thank you.
Thank you, ma'am. A very good afternoon to dignitaries on the dais, participants online and offline. I would like to congratulate the departments of biotechnology, microbiology, and zoology for conducting this kind of conferences. And I want to impress upon the students the significance of these conferences. So you are our direct target. So I want you to utilize this opportunity because this is a unique experience, different from that you actually get during your classroom teaching and your regular textbook syllabi. So here you get to meet new people and you get to interact with the experts in the cutting technologies so that you can improve your network. So networking in the sense that you want to in your professional life also after your studies, if you want to escalate in your professional life, you need this kind of networks, you need this kind of exposure. So please remember that these conferences and seminars that our Government Degree College for Men's Srikakulam is organizing under the leadership of our principal Dr. Rati Suneka ma'am is directly targeting for you. So the students are the ones who have to utilize this opportunity and get the maximum benefit from this. Okay. So thank you ma'am for giving this opportunity.
Thank you, Ma, for your words regarding the students and regarding your work conference. So this is a very hot situation, but be patient. So up to the tomorrow onwards. After that, coordinator of this program, Dr. Y. Polnayed, Captain Dr. Y. Polnayed. Please.
Thank you. Good afternoon, dignitaries, mainly principal of this college, Dr. P. Sudhakar madam, and Professor Vijayan sir, very cordial guest of this college, and one more resource person, Mohan sir, and Vice Principal Sachinand sir, and today's conference convener, Pradeep, and organizing secretary, Harita madam, Dr. R. Harita madam, and my dear faculty and students. It is a good opportunity to all the students to utilize this conference to grab some knowledge regarding recent trends. You know the name of the conference, Emergence of Stem Cell Technology in Biomedical Field. Just now we witnessed that epigenesis and how it plays key role in IVF technology. Sir explained very in a smooth manner one can understand. He came to students level and explained it. I know most of the students got that information. I'm going to congratulate my brother Ravi for arranging this type of conferences tomorrow because tomorrow 2 days conference is, and I would like to congratulate our principal madam for encouraging us to conduct this type of programs for the welfare of the students because she is also appetite for knowledge. Appetite for knowledge is the force behind this. That's why I'm very happy to be part of this program. And thank you very much for make me to be part of this conference. Thank you.
Thank you, Naidu. And I would like to invite convener of this program, so Mr. Pradeep, please give your.
Thank you, madam. Thank you for giving me this opportunity. So first of all, I would like to express my sincere thanks to our beloved principal, madam, because within a short period of time, just 2 to 3 days before only I told, madam, we are planning to conduct the program. So please cooperate. Then, madam, go on like that. You please guide us. So this is like this type of cooperation coming from principal, madam, since her arrival to the college only. So thanks for that, madam. So our beloved blessings and cooperation to our department and to our college also. And thanks to our resource persons, first of all, Vijayan sir. So he is very kind enough. Every time I am mailing to sir, please come to our college, this program, please give your consent. Every time he is patiently is giving to his consent and is also coming to our college and enlightening our students also. So thanks for that opportunity, sir, and also the cooperation, sir, and also Mohanty again, sir. So he gave excellently talked to this stem cell technology and biomedical applications and everything. So I think all of the students get more knowledge about the stem cells and their application. So you don't stop from here. Keep on thinking about that stem cells and these applications, and you do better results in your future examination. So thanks for giving me this wonderful talk, sir, to our students and district of Colombo. And our vice principal, sir, Satyanarayan, sir, thanks for gracing the occasion, sir, and also cooperation. And our beloved brother and our sister. So every time they are giving me the opportunity for theirs, even though they have another work also, but they are here for successful completion of the program. And thanks for both of you, madam, for answer. And not but not least from students. Actually, I am afraid I told you. So whether students will come or not, I don't know. But you attended the event and made success of this event. Thank you for each and every student of our department of biotechnology, microbiology, and other departments also. Thanks for your cooperation and support. And I expect in future also I need this type of cooperation from your side and all the faculty side. So thank you for giving me this opportunity. And one thing I want to tell to you is why we are conducting this much programs in our academic year. For example, if you see final year students within these 3 years, the final year students may come across 5 to 10, at least 10 programs every within these 3 years actually. Because not only your final year student, every final year student get this type of come across this at least 10 events during this BSc period. Why I am conducting this much events? Because whenever you go to any hotel or any restaurant, what we are asking, please give me menu. Menu means what are the different items they have. Based on that, we can select one item, the best item like that. Like that, in sciences also there are so many items. So the students are confusing why which field is good for. which student and for example one student this field is suited for me. Whenever students join in a BSc program, the students generally ask is, sir, what type of opportunities are there in biotechnology, or what type of opportunities are in microbiology, or what type of opportunities there in zoology, like that they're asking. So it is not possible to tell about that, give overview of these opportunities to the student at that time. So during course of studying these 3 years, we are giving awareness to the different types of program. Now stem cells, one type neurobiology, and another type is plants diversity, another type. There different types of topics we are handling the program. So sometimes I got that during some after some years there is no topic to select the event actually. So this much diversity is there in the topic selection, and also this much opportunity is there. So each one student get one opportunity. Stem cells, stem cell is one opportunity for our students to pursue research, or there is stem cell banks also there. So Srikakulam also recently established stem cell bank. So like that there is opportunities are there. So please select any one of these topics and do your level best and get either entrepreneurship opportunity or job opportunities or research opportunities. So because this is our college and our principal and our tagline motto to introduce each and every field of biology and biotechnology and life sciences. So because based on that, the students will select any one of that and they can pursue their higher education or research career or job opportunity. So thanks for giving me this opportunity, and the DBT and Science Engineering is able for supporting this opportunity. Thank you very much.
Thank you, dynamic convener, Mr. Pradeep. Actually, the biotechnology department is standing in the entire state. It's very proud to feel like because of him. So, PUP is glad because the entire state, knowing the Department of Biotechnology, all are looking to Srikakulam. So biotechnology means Srikakulam, Srikakulam means biotechnology. That is the great of our college department of in charge and coordination with Harita madam also. And there is a second thing, one major also decided by the Commission of College Education in this college next to first year's college, biotechnology is one of the single major. So another two posts will be sanctioned to the Department of Biotechnology. So these are all very because of only I am proudly saying that I am only because of Pradeep. So research actually they gave for the research. Biotechnology means that for research 4 years course. Okay. So please utilize this opportunity and please convey to your juniors also because this is great opportunity. Entire zone, though the two places are announced, that is one Srikakulam and Vishaka Krishna College. So two major biotechnology departments going in the single major program. So all the biotechnology students please utilize that. And also I would like to thanks very thankful to DBT and SAB because they already agree with we are also one of the family members. GD SMEN is one of the family members of DBT and SAB because since from last 10 years they providing all financial, all assessment, infrastructure, everything they are providing us. So thank you so much, DBT and SAP also. Now I would like to Sri Mohante sir, Professor Sri Mohante sir, Assistant Professor from Bangalore. Please give your words. Thank you.
Yeah, so I'm really feeling proud and excited to be here, and I hope you enjoyed my lecture. And I think this kind of activity is very very important to, like, we do a lot of things in the lab, and as a scientist, bring you to the young mind so that they get to know the current update of the modern science, as well as it helps them to be enlightened and excited and motivated, and it will definitely help to choose you for your future careers and for more higher education. So that's why I'd like to thank very much Pradeep and organizing committee and principal and vice principal. And yeah, I'd be happy to come again. So please feel free to call me. I really enjoy. And one thing at the last I want to mention that Yeah, I was seeing all of you at least listening the lectures or both of the lectures. So none of you are busy in the mobile. I think that's a very, very good thing. I think I must extend my thanks to teachers for this. Okay. Okay. So yeah, thank you very much. Yeah.
Thank you, sir. Thank you so much. And again, we will compulsory invite you. Please come and attend so many programs, and we will both together will do, sir. Thank you. Actually, finally, I would like to invite today's program chief guest, Professor Vizian, sir. He is a great personality. Actually, you like 20, 17 years, okay? But you are suffering with some struggle, some weakness, some weakness, and everything. But do you observe him? Do you observe him? So he is very active in these days. So that is very greatness. Where you call them, where you call him anywhere. So he will go and attend the programs, and he is very interested on science interested. So very glad to, very proud about you, sir. Thank you so much for attending here. I would like to invite him to give some blessings from him. Thank you, sir.
Thank you very much, Principal and my colleagues on the stage. I must really congratulate your principal and Ravi for taking all initiative. See, unless you, I always say, unless you initiate by yourself, nothing comes. Nobody brings and give you anything. So you have to, demand is easy, but you have to work for it. That's the hard part. And 5, 6 years ago, I think the first program when Pradeep invited me, I don't know how he had given to meet you. And the very first meeting we had here, I know the restrictions being a government college, government institutions always we have different kind of restrictions and facilities. When you go to private, you have other kind of restrictions, but from outside, everything fantastic. That's a difference. The government institutions from outside and inside doesn't look that glittering and attractive. Okay. But in spite of all those, whether you are in private or government, we have our own constraints always. Within that constraints, Pradeep, I found him so proactive, and I always used to encourage because as a young student like you, when I was my professors, I had great professors, and they always used to encourage the students. So I continue that motto and I do. So if try whatever help Pradeep wants by way of sponsoring projects or by way of getting funds or by way of inviting speakers or arranging meetings, I always have 100% support for him. Because he's with all these things, I mean, he is so. I always tell that I told you to principal also is proactive. So you work for something, you will end up achieving something. You may not reach what you started and particularly support for science, since I have been associated with the National Academy of Science, India. The mandate of the science academies in the country is to promote science, encourage science, and take science to the unreached. So, especially last time, being the independence, Azadi Ka Amrit Mahotsav, Amrit Varsha was there. So, Prime Minister has given that mandate to all the science academies, make which take science to the unreached. So, all the academies have several programs to make youngsters at the school level, college level, and the university level. We whatever proposals they send, we have mechanisms to support it by way of financing, by way of experienced persons or scientists who have been the fellows of the academy or who has non-fellows who has done even work uh, nationally and known nationally and internationally. We stimulate them, motivate them to come and address the students. So place like Srikakulam, it's not that easy to come, and I was telling please don't arrange meetings in May, June, July. I don't know, after March, because this one there, and all that in the right report, sitting, 45 minutes or an hour talk. So that's of course okay. But even in that conditions, what I want to emphasize is we, I decided to come and spend 2 days with you just because of our motto is that to promote science, develop and support for the growth of scientific temper amongst the youth and so with that view, Pradeep has done and your principal has given 100% support. See, this is another aspect. There must be the head of the institution should always support their activity. Whatever activities their junior colleagues take it to them. That's also very important. So unless the support comes, they can't act free. So this you are lucky that your principal is also very, very proactive. And whatever, not only Pradeep, any of colleagues, staff member comes with them, she supports 100%. So it is very essential for the growth of the institution and the proper development of the students. So with these few words, I congratulate Principal and Pradeep and Hemda and all his colleagues for arranging this program and for the successful conduct of this program, and it's online also. That's another advantage. COVID has given us another opportunity to conduct online programs. So some people are also far away getting benefited out of whatever is going on. So I wish all of you good luck. The four things you should remember is I call it, we all learn to read, write, speak, and one other essential thing in our life is to listen. The fourth most important thing is to listen. Unless you have the patience to listen to others, and when patiently listen to others, whether you agree or not, you listen to them, then discuss with them if you have the good opportunity or mind, you can debate with them, discuss with them, or argue with them, or disagree with them. So these four things are very essential. And read, write, and speak, we are taught after we are born. But listening, nobody teaches us. That has to come from you. Okay. So the fourth aspect is very important. It has to come from within you. Nobody is going to teach you how to listen. Okay? It's you have to develop yourself. Then think. That leads to thinking and things. So please keep reading. Don't abandon the habit of reading. Even mobile, if you don't read, I will slowly tell you when I check tomorrow, I will check this one. The reading, the advantage of reading. If by not reading and not listening, I'll tell you a small story. Okay? Thank you.
Okay, sir. Thank you for your words regarding our conference and also heartful words to about our parents and students and faculty also. Thank you. Thank you so much. And so actually, I would like to some words from student side. So students, in this morning, most of the 50 are attending here. What about in the afternoon session? How many students will attend this afternoon session? Please raise your hand. Online. So everybody. So students will also link and online. Okay. Take lunch. So after go and attend anyway in the offline or on online. Okay. Thank you so much. And I would like to invite participation program by the coordinators and conveners to our vision sir.
Thank you, sir. I now request Srimonte sir to accept our small gift of appreciation. And I request all the dignitaries, all the directors to please partake in this. I request all the students to please assemble in the front of the stage for a group photo. Fast, please. 2 minutes. Come fast. Please assemble below the dais. Come fast. తొందరగా రండి అమ్మా. Come fast.
Respected dignitaries on the dais, respected principal ma'am of our college, Dr. P. Surekha Garu, and today's chief guest, Vijayan sir, and Mohan sir, and vice principal sir, Dr. Coordinator Garu, and program of this coordinator of this program, Y. Coordinator Garu, and Artha Madam Garu, and convener of this program, M. Pradeep sir. Very good afternoon to one and all, and very good afternoon to my dear students friends. ఈ కాలేజీలో నేను ఎన్నో ప్రోగ్రామ్స్ అటెండ్ అయ్యాను.
మీ మీలాంటి పిల్లల్ని ఉద్దేశించి ఈ కాలేజీ చేస్తున్నట్టుగా మన స్టేట్ లో ఏ కాలేజీ కూడా చేయట్లేదు. యు ఆర్ వెరీ ఫార్చునేట్. గవర్నమెంట్ డిగ్రీ కాలేజ్ మెన్ శ్రీకాకుళం అనగానే ఒక బ్రాండ్ అంబాసిడర్. ఇక్కడ చేసినటువంటి రీసెర్చ్ యొక్క ఓరియంటేషన్ ప్రోగ్రామ్స్ కానీ, సెమినార్స్ కానీ, కాన్ఫరెన్స్ కానీ, రిఫ్రెషర్ కోర్సెస్ కానీ ఏవైనా సరే ఈ కాలేజ్ లో చేస్తున్నట్టుగా ఇంకెక్కడ కూడా చేయట్లేదు. సో యు ఆర్ వెరీ వెరీ ఫార్చునేట్. సో టుడేస్ టాపిక్ ఈస్ ఎమర్జెన్స్ ఆఫ్ స్టెమ్ సెల్ టెక్నాలజీ ఇన్ బయోమెడికల్ ఫీల్డ్. ఈ టాపిక్ అనేది మెయిన్ గా బయోటెక్నాలజీ వాళ్ళకి, మైక్రోబయాలజీ రిలేటెడ్ గా ఉంటుంది. ఎక్కువగా స్టెమ్ సెల్ టెక్నాలజీ అంటే రీసెంట్ అడ్వాన్సెస్ కి ప్రెసెంట్ టెన్స్ కి ఎక్కువగా ఉపయోగపడేది ఏంటంటే స్టెమ్ సెల్ టెక్నాలజీ. ఈ స్టెమ్ సెల్ టెక్నాలజీలో మనం వచ్చేటటువంటి అడాప్టేషన్స్ ఏంటి, మోడిఫికేషన్స్, వాటి యొక్క టెక్నిక్స్ ని మనం నేర్చుకొని ఫ్యూచర్ లో వాటికి తగ్గట్టుగా మనం తయారు కావాలి. సో ఈరోజు చెప్పినటువంటి మనకి చీఫ్ గెస్ట్ విజయన్ సార్ గారికి చాలా మంచి సందేశాన్ని అందించారు. అదేవిధంగా మోహన్ సార్ కూడా ఎంబ్రియోజెనీ అండ్ ఐవిఎఫ్ ఇన్విట్రో ఫెర్టిలైజేషన్ సంబంధించి మంచి టాపిక్ కూడా మనకు అందించారు. సో వాళ్ళిద్దరికి నా తరపున, కాలేజీ తరపున, మీ అందరి తరపున ప్రత్యేక ధన్యవాదాలు తెలియజేసుకుంటున్నాను. సో ఈ కాలేజీలో మీకు ఇటువంటి కాన్ఫరెన్స్ కానీ, సెమినార్లు కానీ ఎన్నో జరుగుతూ ఉంటాయి. సో స్టూడెంట్స్, మీ ప్రిన్సిపల్ మేడం గారు నిజంగా మీ అలాంటి మేడం గారు దొరకడం మీ అదృష్టం. నిజంగా అండి, మీరు ఎంత కోఆపరేట్ చేస్తున్నారంటే నాకు తెలుస్తుంది. ఎన్నిసార్లు వస్తున్నారు, అటెండ్ అవుతున్నారు కాబట్టి. అంతగా కోఆపరేట్ చేసే వాళ్ళు ఎక్కడ ఉండరు. సో అటువంటి మేడమ్స్ కానీ, అటువంటి ఫ్యాకల్టీని మీరు యూటిలైజ్ చేసుకొని మీలో ఉన్నటువంటి స్కిల్స్ ని డెవలప్ చేసుకొని వాటిని బ్రైట్నెస్ చేసుకొని ఫ్యూచర్ లో మంచి స్థాయికి ఎదగాలని కోరుకుంటూ నాకు ఈ అవకాశం ఇచ్చిన అందరికీ మరొకసారి ధన్యవాదాలు తెలియజేస్తున్నాను.
Thank you, sir. With this, we are closing our morning session, and I request all the participants, students, and our delegates to please join us in our lunch in Silver Jubilee Auditorium. And post lunch, we'll be posting our link for the afternoon session. Please join in the link. And tomorrow morning, I request all the students to please reassemble here in this hall at 9:30 a.m. And the certificate distribution will be done tomorrow itself. Thank you.
Dear online participants, this is the time for lunch. From 2:30 onwards, one more online session is going to start. Thank you.